my lab in Oregon. North Carolina, we have an entomologist there that worked with us. Arkansas, one of my former students, now has Grigorovich's position. So Yanis is involved, and then a colleague in Mississippi. And we started looking at this blackberry decline, or blackberry yellow vein, in the southeastern U.S. And we found plants that were very, showed those symptoms. We actually had blackberry plants that were dying in Arkansas and the Carolinas, and they were testing negative for the 4 common viruses that we knew of in blackberries at the time in North America. So there's something else going on. We did electron microscopy with a group in Arkansas, and we found pinwheel inclusions suggesting, okay, that shows us it's a potyvirus. There's a potyvirus there, which is a large group of plant viruses. We did double-stranded RNA. When you find double-stranded RNA, you don't find that in healthy plants. It's only in virus-infected plants. So it's a really good way to look for viruses. And, electron microscopy, it's a virologist screen. Things are looking good. So these are the pinwheel inclusions. That tells us it's a potyvirus. These are dsRNA bands here. 1, 2, 3, 4, 5, 6, 7, maybe 8. So suggesting multiple viruses. And these are just molecular markers. But this suggests that we're looking at a virus complex. And then we went in and we extract that dsRNA, we clone it, sequence it. And at that time, this is what we were doing. We were doing dsRNA shotgun cloning. Now what we would be doing is we would take that dsRNA, do next generation sequencing on that, or take total RNA from the plants or small RNAs. Then we also do rolling circle amplification because that's specific for DNA viruses. The dsRNA we won't find from a DNA virus. So we do this as well. And applying that, at the end of the day, in Arkansas, we have these 5 viruses. Blackberry yellow vein and blackberry clotted ring spot are the 2 most common. The potyvirus, when you see potyvirus, the first thing you think of is aphid transmission. So once we saw the pinwheels, we started doing aphid transmission studies, and we could never get it transmitted using aphids. But once we had the sequence information, we knew that it was missing these 3 essential amino acids that are necessary for aphid transmission. So, okay, it should be aphid transmitted once we have that sequence information. The tobacco ring spot, which is nematode transmitted, and then flexularity. So that's Arkansas. In the Carolinas, again, we have blackberry yellow vein, we have blackberry clotted ring spot, and here, tobacco ring spot is very common. But INSV and beet sooty yellows were unique. We did patients with chronic shock and beet sooty yellows. We only saw those in North Carolina. Then we go to Mississippi, but symptoms still look very similar. Go to Mississippi, we have blackberry yellow vein, but the other 4 viruses found in Mississippi were different. So what's happened is that in the late 80s and 90s, they started producing a lot of blackberries in that part of the country. And they hadn't grown very many blackberries there before. There's probably the viruses in the native vegetation are now coming into blackberry, and that's why we're seeing all these new viruses. If I was a betting person, I would say you're going to see the same thing here 10 or 15 years after the berry production has exploded. And the berry production has exploded here. It's become a huge industry. So I'm expecting that you're going to see new viruses here that we haven't seen in these crops over the next 10 to 15 years. You probably have different vectors. White flies are much more common. We've got a lot more viruses in the Gemini viridae, small group of small DNA viruses that we don't see very many in the berry production areas where we are. So I'm expecting that 10 or 15 years, there'll be a whole list that'll be unique to Mexico. That's a guess. So, symptoms were similar, but the virus complex, the virus mixture, is different. And none of these viruses, I think we have 13 or 14 now from those blackberries, cause symptoms in blackberry by themselves. So if you just look at a plant that has any one of those viruses, it looks healthy. The more viruses you have, the more severe the symptoms. And there's no virus that was present in every symptomatic plant. Blackberry yellow vein and blackberry clotted ring spot are the 2 most common, but we have a lot of plants that show these symptoms that are negative for these 2 viruses. So it's always a complex, and there's not a specific one that's critical. But if you go to Arkansas, these are the two most critical. If you go to North Carolina, these two are critical, but also tobacco ring spot. You go to Georgia, Mississippi, this is probably the most critical. So then you could start planning a strategy for control. This one is white fly transmitted. Tobacco ring spot is nematode transmitted. You can start trying to figure, okay, if it's white fly transmitted, what can you do? Nematode transmitted, and start trying to develop a strategy. So these are the different viruses that we've isolated from plants showing black-gray elevation disease. And you see nematode vector, pollen vector, unknown, thrips, erified mites, and white flies. The problem with erified mites, thrips, and white flies, they move a lot. So these things can be difficult to control. And if you only have, black-gray elevation, and you don't have these others that are transmitted by thrips or erified mites, you have one virus that you need, one vector would be the white fly. And the INSV, the invasive zoonotic spot, is really we've only seen that in North Carolina. So at this point, that's the only place that's an issue. Nematode transmission, it's got to be a pre-plant decision, because once the plants are on the ground, there's not much you can do for nematode control. So you need to do nematode surveys ahead of time, and if you have the vector and the virus, you need to go in there and do some type of nematode management program before you put the plants in the ground. If you have the nematode, but no virus, it's not going to be a problem. Because these nematodes, the Zithonema, the populations I've never seen them high enough in raspberry or blackberry to cause a problem. They're only problematic if they're transmitting virus. So if you don't have virus and you have nematode, you can survive that pretty well. So that's part of a big project that we did over about 15 years in the southeastern U.S. And I want to talk a little bit about a crumbly fruit disease that we have in the northwestern part of the U.S., and that's where I am. And both projects were funded by SCR, which is Specialty Crop Research Initiative. It's a new funding program that started back in 2008. So we've been able to get funding, large-scale funding for these. But with the crumbly fruit disease, it really became much more important in the 1990s, associated with a shift in raspberry cultivars. Prior to that, most of the cultivars that were grown, or the dominant cultivar, was resistant to raspberry bushywort. Meeker, and the other thing we've noticed is that even this cultivar, the same cultivar up in northern Washington and into British Columbia, Canada, the symptoms are much more severe than they were in Oregon, which we're about 500 kilometers south. And we have the same virus, but our symptoms are much milder on that cultivar. And similar to the situation with tobacco ring spot, at the time, raspberry bushywort is the virus that we could get out of these plants. That's the one that we could mechanically transmit, we could detect, and we knew it was there. But then, since we saw these differences between Oregon and Washington, okay, there's got to be something else going on. And this is a raspberry field up in northern Washington. It looks really, really good. And these viruses, even if you have 3 or 4 in there, the plants look good, but the fruit is very crumbly, and the production will go down. But the plant itself looks quite healthy. There's a breeding program up there, and we started walking through their plots looking for indication of virus, something that would suggest virus. So we're walking through these plots, we see these symptoms, and we tested for these are the common 3 viruses that we might see up there. Tomato ring spot's not common, but it's one that we know occurs, and this one is more common in Oregon than northern Washington, but this is the major one. And so these plants were negative for the 3 viruses we knew about. Extracted dsRNA, cloned and sequenced, just like, and we found raspberry leaf model, which is a typical cluster virus. So cluster viruses are all transmitted by aphids. So once we had this virus, we did some aphid transmission studies using the raspberry aphid. There's really one aphid species that we find common on raspberries, and sure enough, it transmits it very well. Raspberry latent it's a reovirus. And at the time, all known plant reoviruses were transmitted by leafhoppers or plant hoppers. So that was going to be our guess. That should be our best guess for the vector here. I'll talk about the work we did on this one because it was a surprise. Because based on the sequence, our first stop was leafhoppers. And then we did rolling circle amplification to look for DNA viruses. We found Rubus yellowman virus, which is a panthervirus and aphid translator. So the raspberry latent, which is the unusual one, so it's got 10 pieces of RNA, double-stranded RNA, and 2 of those code for multiple genes. So there's a total of 12 genes, and these black pieces here are inverted repeats. So in nature, those things would form a circle. I mean, we draw them on here like this, but if you put these in solution, these 2 pieces here will hybridize each other, and each one of these will be a circle. And this end, the ends are the same on all of these. So what we get, this is the section that hybridizes, that varies back a little bit. These are the 2 ends. And we suspect that this is important because this virus particle, to be infectious, all 10 pieces have to get in there. And it can't just be a random event. So these are probably involved in assembly so that when the particle is formed, all 10 pieces are there. We actually have an infectious particle that's going to move to the next plant and multiply. And this is raspberry latent, and the closest virus to it is rice ragged stunt. This is an interesting one because this is actually mosquito virus, which is the second closest, most closely related virus. And gene number 6 from this virus is most closely related to a gene out of this mosquito virus. It may also explain a little bit about what we're going to find with transmission, because mosquitoes feed very much like aphids. They've got a pierce, they pierce in, they suck out. Aphids do the same thing in plants, mosquitoes do that here. So anyway, this was surprising, but in retrospect, maybe it makes some sense. So we have these viruses, then we did several studies to look at when do they move. Because if you tell the grower you've got these viruses that are aphid transmitted, their first response is to go out and start spraying for aphids every 2 weeks or whatever it's going to be. So we did a number of studies looking at, so this is we represent a field. We put out 16 trap plants, and the trap plants, we'd take them out, put them in the field. 2 weeks later, we'd bring it back to the lab and take another set out. And we did that every 2 weeks for the entire growing season. And at the end of that, we can test every group of plants so we know when the virus is actually moving. If we don't have any movement in May, okay, if none of those plants become infected in May, there's really no reason for a grower to think about spraying because the virus isn't moving. And if it get a lot of movement in June, okay, that's the time to control, and maybe very little movement later on. So we can tell the growers, this is what you need to do to try to control this disease. And then the other thing, these yellow bars represent sticky cards. So we're also looking at aphid populations, because in this particular case, we know it's an aphid transmitted virus. So we put plants out and bring them back to see when the plants are becoming infected, sticky cards to see when the aphid populations are looking like. And so these are the sticky cards that just kind of post in the middle of the field. Trap plants go right in the row of the field, and the growers can run their harvesting equipment over this, and these are right in the middle of the row, it doesn't interfere. And 2 weeks, this will be out in the field for 2 weeks, then come back. And this is what we found. Basically, no transmission until the very end of May. And then these are the number of positives. Most of the transmission is occurring in June. The aphid populations really take off here. And then, so the biggest aphid flight is during the month of, this is in Oregon, during the month of June. Relatively few aphids and very little transmission in July and August. So we can tell the growers, okay, if you want to control this, you want to put maybe a spray on right at the end of May, because that's when the aphid flight hits, and you might be able to do that with 1 or 2 sprays. Just knock this peak down and try to get rid of it. So that's the type of information we're trying to capture by doing these field part of the study. And that was in Oregon. Now we did a similar thing up in northern Washington. And so we put out, in this case, we did yellow pan traps as well, because that will get aphids that are flying over the top of the canopy that might come down, and you get a lot more species of aphids collected. And also sticky cards. We put those out, and we focus on raspberry leaf model, which is one that we know is aphid transmitted. We focus on raspberry latent, which should be leafhopper transmitted, and we're also looking for leafhoppers on these sticky cards. So this is the leafhopper that's common in raspberries and blackberries. This is the aphid, and so the latent, this is our best guess because they're all leafhopper transmitted, and this we know is a vector for leaf model. But one thing that doesn't quite fit is that raspberry latent is much more common up in northern Washington at the Canadian border than it is for us in Oregon. This vector, we see a lot more of this in Oregon than we do up north. So we're basically looking at both of these as vectors. And Diego Quito worked on this. He's from Ecuador. He did this part of the project. And so what he wanted to do was he wanted to create mixed infections, but he also wanted to look at the vector. So we had a lot of plants that had mixed infections. He's using aphids to try to get a plant that we can singly infect with raspberry leaf model. And out of one of the 9 plants he inoculated with aphids tested positive for raspberry latent virus. And so I told him to go back and try that one again. And he did. And he was right. But the other thing he did, when the aphids feed on that plant, this is a real-time PCR. So at day 1, when the aphids are feeding on there, you get, the further the line moves this way, the more virus that is present. So day 1, and that they fed for 1 day, and then they were put on healthy plants. So they're not taking up any more virus, but the virus continued to increase inside the aphid, even though they're not feeding on infected plants. This shows that the virus is actually multiplying inside the aphid. And then what he did was, so at day 1, we can detect the virus. In 5 days, it goes up significantly. But then it starts going down again. And what happens about this point, days 5 or 6, the aphid can start transmitting. So with the raspberry latent, with the raspberry leaf model, the cluster virus, the aphids pick it up, and in a couple hours they begin transmitting. With raspberry latent, they feed on a plant, and it takes 7 or 8 days before they can transmit. That makes a big difference as far as management, because this virus is going to move much more slowly. And the other thing with this, the fact that it's going down suggests that maybe the aphids can be able to transmit efficiently for a while, and then the efficiency is going to go down if it's losing virus once it starts transmitting faster than it's multiplying. So this is the transmission experiments. So if he put the aphids on the plant for 1 day, he was not able to get any transmission until day 8. 2 days, day 8. 4 days, day 6. Well, the thing is, they're picking up the first day here already. So basically the latent period, the time from starting to feed before they can actually infect a plant, is somewhere in the 7 or 8 day range. That means if you put a spray on today and a new aphid comes in, you don't have to worry about that aphid for at least a week. So you adjust your spray schedule if this is the virus you're trying to control. And then he did transmission studies. The other thing he found was that you had to use a lot of aphids to get this thing transmitted. With raspberry leaf model, 3 aphids, you get a high percent transmission. With raspberry latent, you had to do about 50 aphids to get a few plants transmitted. So it's very inefficient. And again, that means this virus should be much easier to control than raspberry leaf model because it doesn't move very fast. You need high populations. All things that are come into trying to develop a management strategy. And then we have an entomology student, Danny Lytle, that looked at the aphid biology and basically using the Grenier model, looked at aphid movement with traps when she was doing the trapping. Now up in Oregon we saw transmission, we saw aphid movement about the 1st of June. Up at the Canadian border, it's almost the end of June. So we can tell the growers, don't even think about spraying for aphids up there until late June because they're not moving. You know, if you're spraying in May or early June, you're not doing any good. So the growers now can move this, you know, they don't have to worry about this spray. And normally what they do is they do what they call a cleanup spray. So about 10 days before harvest, they would go out and spray for insects so that when you're harvesting fruit, you don't have aphids on the berries. Most people don't find that very attractive, and they're hard to get rid of. So they would go out and normally do a cleanup spray. So what we're doing is we're working with a number of growers now on 20-acre blocks where they're taking this cleanup spray and moving it here, moving it up 10 days. So they're not doing any more sprays, they're just changing the timing to see if we can knock this aphid pop, you know, hit these aphids right as they start building and control virus spread without change, no more sprays, just adjust the timing. And we have another set of growers that are doing, actually adding one extra spray here to see if that might increase the efficiency of controlling these aphid-borne viruses. And these are 20-acre blocks, and these are long-term studies that are still going on. So we're trying to come up with a strategy for the growers to say, okay, if you move this cleanup spray 5 days, 8 days, 10 days, you're going to get good aphid control as well as cleanup. No extra cost. Aphid and virus control. So the graft inoculations, you know, Diego had these, and then he made all various combinations, and he set up some field trials. And this is the healthy, every single virus-infected plant, no matter 1, 2, or 3, established poorly. Now this is the healthy, everything's established poorly, so we plant in spring, they're not producing any fruit that year. In the second year, the growth was fairly good all the way across, except for we had bushy D as bushy worm and leaf model. That seemed to suppress growth. And when it came to fruit quality, it's bushy worm and latent seemed to have the biggest impact on fruit quality, just getting poor berry set. So we get berries like this, that's juice, jam, and yogurt. This berry can be in the fresh market or the whole frozen berry. This is much more valuable. Growers will lose money if that's all they can grow. They don't make enough money to pay for the operation. So this is what they're after. So controlling leaf model improves plant growth, especially establishment. Controlling latent improves fruit quality. So the other one that we're concerned about is, so we can try to spray, but there's a lot of work been done with aphid resistance. How does aphid resistance affect the aphid feeding, and how does it work? Are there differences? So we have aphid resistance in red raspberry, we have aphid resistance in black raspberry. If they behave differently, can we put those together and get something that's going to work a lot better or work a lot longer before it breaks down and have more endurance? So the entomologist in Gang Lytle started working with virus-infected plants, and aphids lived 30% longer on virus-infected plants than they did on healthy plants. The average is 27 up to 36, so 30% longer lifespan for the aphid if they were feeding on a virus-infected plant. The other thing that's really striking is they produced twice as many young on virus-infected plants if they had these two viruses than they did on healthy plants. So then the other thing Danny wanted to do was, okay, when are they feeding? And how do they react differently to an aphid-resistant plant to an aphid-susceptible plant? So she wired up the aphids. So this is the aphid, little gold wire, and a circuit comes into the pot, and the other wire comes out here into the soil. This one comes up to the aphid. So the aphid probes and actually probes into the plant. It completes the circuit, and you can measure that voltage. Here's the aphid. You can't really probably see that little fine gold wire there. But the aphid's free to walk around here. They survive very well. They'll spend days on here. And then you can monitor their feeding. And what you found is that in a susceptible plant or a resistant plant, this is when the aphid is salivating. As the aphid's taking its silent, the aphid is looking for the phloem tissue, the vein, so it can suck out the phloem contents. And as it's going in, it's spitting out or ingesting as it's going in. And then once it gets to the phloem, this is on a susceptible cultivar, it starts sucking up the contents. Well, in a resistant cultivar, this is a perfectly flat line. It can't suck anything up. This looks exactly the same, but on a resistant cultivar, nothing happens here. So on the resistant red raspberry, the aphid can't take anything out of the phloem, so it leaves and it goes to another plant. So you come back the next day, if you have a susceptible and resistant plant, all the aphids are on the susceptible plant because they've tried this, they can't get anything, they eventually move off. And when we have that aphid resistance working in the field, we see very, very little virus transmission. And the other thing we had an entomology student with one of our entomologists. Michael Dossett was a graduate student in plant breeding, working with Chad Finn, the plant breeder group, and I had Alfredo and Diego had two graduate students working in virus. So we had a really good graduate student team there. And Michael Dossett identified aphid resistance in black raspberry. So then Danny said, okay, do they behave the same? So in red raspberries, aphids are not able to suck up out of the phloem. In black raspberries, aphids be penetrating, they would get to the phloem and they would just keep on going. They didn't realize they were in the phloem. They could never sense that they were in the phloem, so eventually they would actually go into the susceptible plant. So there's two different mechanisms. One that seems to coagulate, that they can't suck anything up, and the other one they can't recognize. So Michael Dossett now is a plant breeder up in British Columbia, and he's working hard to combine these two into a single plant. So we have two different mechanisms of aphid resistance, with the idea that it would last a long time if you use that in the field. So that was a really fortuitous thing. We had 4 graduate students all working together on various aspects of this problem. And then looking at, I said raspberry latent should move much slower. This is up in northern Washington, right along the Canadian border. And on 5-year-old fields, you see a lot of these are 100% infected with raspberry leaf mold. But even at 6 years, we have the one field here, or 2 fields that are very high. But raspberry latent moves much, much more slowly. And that's what we'd expect based on the work we did in the lab, and that's exactly what we see in the field. And then in southern Washington and Oregon, we just get very low aphid numbers compared to what they see up north. And even raspberry leaf mold moves much more slowly for us than it does up north. So we don't have that same disease pressure that they have. Our fields last a lot longer and still produce high-quality fruit. Up in northern Washington, they're replanting about every 5 years because of crumbly fruit. So this is northern Washington. This is where we have very high aphid populations. In this area, southern Washington, we get very low aphid populations. Our yields are much lower. So even diseased plants up there, they can produce 10 tons per acre. On our really good fields, we get 10 tons per acre, but it's high-quality fruit. On healthy fields up here, they should be able to get about 18 tons per acre, and that's why at 10 tons per hectare, they're not making money. So that's why these fields are being turned over so quickly. The other thing we have, common in all these, is raspberry bushy-wort. Now, raspberry bushy-wort is pollen transmitted. So, okay, that's difficult to control because if you prevent pollination, you're not going to have fruit. But we've had resistance to raspberry bushy-wort in a lot of our, in our major cultivar prior to 1995, the cultivar Willamette was resistant. And now most of the new material that's been introduced since then is susceptible. And that's why we're seeing a lot more raspberry bushy-wort than we used to 20 years ago. Well, the breeding programs from 1995 until now have really focused heavily on taking this resistance gene and getting it into new cultivars. Well, in 2014, so 2 years ago, we found a strain of raspberry bushy-wort that infects, that overcomes that resistance gene in Washington. So then the question was, if you have a raspberry plant, the cane comes up, next year it's going to produce fruits or produce a fruiting lateral, you've got flowers out here, that's when pollination occurs, so that's when the virus is moving. But during fruit development, this should be a sink. So if everything's coming this way, the question was, at harvest, if you cut that cane off, has the virus been able to move down to the root system when you have the sink up here? So that was a question the growers had. So as soon as the harvest is finished, cut the cane off. If the virus didn't make it into the root system and you cut that cane off, you don't have a systemic infection. So that was one of the things that a graduate student with Pat Moore was a breeder up in Washington. We worked with them on this. And then the other question was, do mixed infections result in more efficient pollen transmission of raspberry bushy worm? Just a question that we don't have an answer for. The RBD infection via pollination. So what Para did is have a beehive in the. So she had every third plant was RBD, had bushy worm, and then she introduced the beehive into the greenhouse or screen house, and then had the pollination go. 1 week later, 2 weeks later, 3 weeks later, she started collecting various tissues. And at the end of the first year, everything was infected, and I said, there's got to be contaminating pollen or whatever, because it can't move that fast. So she did it again, and a lot more care and detail. And what she's finding is, so what she did then halfway down the cane and at the base of the cane. And she started doing that 1 day after pollination. And what she found was 1 and 2 days, the receptacle. So even 1 day after pollination, the virus had moved from the pollen into this tissue. And that just really surprised me. I had a hard time with that one. And the pedicel, most of the pedicels there already had virus. The fruiting ladder, half of them, the virus had moved into the fruiting ladder. And the base of the cane, all the way down to the base of the cane, small numbers because it was a destructive assay here, but for 3 and 4 days, the virus is at the base of the cane. So what that tells us, early cane removal is not going to help bushy worm control. So we can tell the growers, getting those people out there, getting a labor crew out there when they should be picking blueberries or something else is not a good idea. But that's information that we can now use to tell them. And the other question I asked was, do mixed infections influence the rate of pollen transmission? And the reason we asked that was, this is looking at the titer of raspberry bushy worm using real-time PCR. And this is bushy worm by itself. And this is bushy worm in mixed infections with raspberry leaf mold. And what we have is about a 400-fold increase in the amount of virus. Not a 4-fold increase, 400-fold. So if there's that much more virus in the pollen, his pollen transmission would be much more effective. And that's one of the things we're trying to work on now. And this is real-time PCR. This is conventional PCR. Bushy worm by itself, mixed infections, 15 cycles, really bright bands, really faint bands. ELISA, mixed infections, single infections. So, the ELISA test and the conventional PCR all point in that direction, but the real-time PCR do exactly the same thing. So that's why we're now asking the question, having that leaf model there, is that going to impact the rate of bushy worm spread? Rubus yellowhead, this is a project that Alfredo Diaz-Lara from Veracruz here, well, not here, but the other side of the country, is working on. He's doing his PhD, just hoping to finish here before Christmas. And I think he's on track to get that done. So, Rubus yellowhead is a DNA virus. It's transmitted by the same aphid, and it's, in a mixed infection, it contributes to this mosaic disease and poor fruit quality. These are the type of symptoms we get on indicator plants. This is black raspberry. These are detection. Traditionally, we always detected this by grafting based on symptoms. Once we had a PCR test, what we found is that we had a lot of plants that were negative in graft indexing that were positive by PCR. which didn't make a lot of sense. So we did grafting with these, and we take that, after we do the grafting, we test the indicator plants. They're negative by PCR. So it's not just that they're not showing symptoms, there's no virus being transmitted. And so it's not like we have a mild strain of the virus that we're missing with graft indexing and positive by PCR. It's this thing is just not moving. And that was a little bit of a surprise. So turns out we had a number of plants in our virus-free collection that once we went back and tested by PCR, they're PCR positive, but they're all graft negative. And so then we started looking for one that was graft positive and PCR positive. And we ended up settling on this one, which we got out of the germplasm repository in Corvallis, originally came from Scotland. And did rolling circle amplification, and we were selling it. When you do the digestion, this is total, this is after digestion. This shows that you have a DNA, most likely you have a small circular DNA. Good chance it's virus. And Diego took that, cloned it, and sequenced it, and this is what he found. And this is a published sequence from Canada, and you'll see in this one there's 7 different genes. Here we have 3 complete genes and a couple of partial genes, but it's quite different. Gene number 7 is missing. Gene number 5 is missing in the bumper seedling. Gene number 6 is short, much shorter. So, but genes 1, 2, and 3 are very, very similar. And those are the 3 classical or genes that you always find in adenoviruses. But it's surprising that we have these 2 that are, based on the RNase H, which is the most conserved region, they're very, very close. But then the fact that you're missing all of this genetic information is surprising. This is the Mumford seedlings. That's the Canadian one looking at the RNase H. That's the most conserved part of the virus. But these 3 always cluster together. So the question then is, do we have virus particles in some plants? And the other plants, is that virus sequence actually part of the incorporated into the raspberry DNA? And that's what is happening. So if we take plants that are graft positive and PCR positive, we get rolling circle product. If it's graft negative and PCR positive, we don't get an amplification by rolling circle, suggesting there's not that small circular DNA present. If we take this plant that's graft negative, PCR positive, and cross it with one that's negative, approximately half of the seedlings are PCR positive. And none of them are graft transmissible, suggesting that it's behaving almost like a single gene. It is behaving like a single gene. And if we take 2 of these and cross them, we get, I think this was 73, but pretty close to the 3-to-1 ratio. And again, behaving very much like a single dominant gene. And when we take these seedlings, we tested a number of these, and none of them are graft transmissible. None of them are RCA positive. And again, suggesting that this particular virus can be incorporated into the plant genome. And Judd, the guy at Driscoll's, Judd, he's a molecular biologist, but he's been doing sequencing of raspberry bushy-bark about the time we were doing this. He called me.
Ding?
Pardon?
It's Ding? Ding?
No, Judd. He's at the Driscoll in Watsonville. But he was sequencing raspberry genome for their breeding program at Driscoll's, and he called and said, we're finding some really weird stuff. It looks like raspberry Lewis Yellowneck virus in everything. And so we started comparing the stories, and it turns out they're seeing the same thing in their breeding line, in some of their breeding lines that we found. They found it strictly through deep sequencing. We found it through biology. It came together very nicely. And so this incorporated component looks very much like the material that's in the Canadian strain, And so we had the Bamford seedling strain, which we had, that came from Scotland, a very different, one had 5 genes, one had 7. Okay, we've got a North American group and a European group. Well then, when the APS meetings were in Minnesota a few years ago, I went over to Wisconsin and spent a day looking for wild raspberries, and I found rubus yellownet virus in wild raspberries in Wisconsin, and it turns out this is very much like the Scottish strain. So, and that's wild raspberries grown in a state park, not close to any commercial production, so very likely a native component. So this is the idea of a North American and a European clade of groups. Probably is not the case. And just this saying that the incorporated sequence that we find looks very close to the Canadian sequence. So that's raspberry, crumbly fruit, blackberry, as we talked about. Now, when we talked about the viruses in that blackberry yellow vein, there was a whole list, a lot of different vectors. The nice thing here is aphid, 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 aphid. So we have one vector to worry about. We know that they don't really start spreading until you're up north, late June. Okay, from the grower's point of view, I can sit back tight. I can maybe put on one extra spray, and probably get pretty good control. And that's what we have a number of growers looking at that right now. Tomato ring spot is rare for us in the Northwest. We don't really worry about it. We suggest growers test their fields before they plant. If they find the nematode, we will go in and test those nematodes for virus. But that's not a common problem for us. But so for our situation, one vector, much easier to deal with than what they have in the Southeast. And this is just, so we set up with growers large blocks to look at spray programs. So blackberry yellow vein disease occurs here. The crumbly fruit complex is here. When we talk about nursery surround, we don't want to move infected material from here to here or there to there. Strawberry. So I'd say a little about strawberry. This is Totem, cultivar from British Columbia. They use a matted grow system. It's very different from what you do here. The plants are put in the ground. They're there for 3 years. Judd Warren. Yeah. So strawberry decline. In California, they had a problem that was costing about $25 million in the Watsonville area in 2002 and 03. And they saw it again in 13 and 14, more on Santa Maria. And then 2013 to 15, they had a huge problem in the eastern U.S. with plants that were coming out of Canada. So strawberries, we have aphid-transmitted viruses, 5 of them, that are probably the major viruses in strawberry worldwide. And they're transmitted by the strawberry aphid. That's the primary vector. Now, the nice thing about these viruses pretty much only go to strawberries. This aphid, so these should be relatively easy to control. In California, 2003, it's about the time we first sequenced strawberry polydosis, and I was down there, and every single leaf I turned over had white flies. We don't see white flies where we are. And so we had this sequence, and once we had the sequence, it fell into this group of viruses. These are all white fly transmitted, so that was our best guess. And we found both of these in strawberry, and then working with Bill Wintermantle down in Salinas, he had white fly colonies, and sure enough, they're both transmitted by the greenhouse white fly. Now, the greenhouse white fly, it's got a broad host range. It's moving all the time, year-round. So it can go to vegetables, it can go to weeds, it can go to strawberries. So this thing is moving, and it's very difficult to control. So if you have your choice, and you've got both, you want to target the aphid. So once we started looking at this, prior to the work that we did on palidosis, we didn't know of any white fly transmitted viruses in strawberry. So there was no reason for nurseries to try to manage white flies because, okay, we have a few white flies, they're not a problem, they're not a vector, it doesn't really matter. So, but once we had this information, we worked with the nurseries and they implemented a white fly control program, and which also resulted in better aphid control. And so in 2000, so the big problem was in 2002, 3, and 4. In 2005, we went up to the nurseries, in high elevation nurseries in California. We tested 400 samples for 9 different viruses. So 3,600 tests, we had 2 positives. So they did a very good job very quickly. We didn't see aphids, we didn't see whiteflies, and so we would expect very little disease to show up in the production fields in 06. And this is what we saw in 06. And we looked at 4 fields in the Watsonville area. They were planted about the 1st of October. We had 1 positive in December, whitefly transmitted. By April, we had a few more. We have a few aphids. In July, by the end of the growing season, at this point the plants are coming out. There's no symptoms in this planting because there might be a few with mixed infections, but there's not enough virus in there to actually cause a problem yet, even though the growers did not use any insecticides. So if they start clean in the Watsonville area, the natural vectors are relatively low. And if you've got a strawberry-free period, so they take them out the 1st of September, plant the 1st of October. Remember, that aphid only feeds on strawberry. So if there's no strawberries there for a month, the aphid's going to die. So you start clean. So this actually works very well for their system as long as you have that aphid-free period. Part of the problem is now we have people that want to plant in July because the price for strawberries is really good in November, December, and they can start harvesting. But now you've got rid of that strawberry-free period, and you've got virus moving from one crop to the next. And that has caused a problem for them in 2015. This is a situation that happened up in Canada. This is a strawberry field infected with multiple viruses. The reason we got involved in this is the planting of strawberries in Florida in January of 2013. 100% crop loss. They didn't pick. They did go through and pick a few berries, but I mean that's not what they grow strawberries. And so we got involved. This is a field this came from one nursery in Nova Scotia, Canada. This came from another nursery in Nova Scotia, Canada. And it's just right to the line. So we went back, that's why we got involved in working with the nurseries. And the nurseries, most of the strawberry plants that are produced and grown in this part of the country, a lot of those plants come out of here. And the nurseries are up in this area. So a couple of nurseries in Nova Scotia just had a serious breakdown in their production system. And there were about 25 million plants that came down here that did not produce fruit in 2013. And it's a result of strawberry mottle yellow edge and strawberry mottle, both aphid-transmitted viruses. They haven't had a problem with viruses for a long period of time, and they pretty much forgot to really worry about controlling aphids. And it just got away from them. They had a blow-up, and one of the nurseries is out of business. The other one is working hard to get producing quality plants. But that's a situation where the nursery management becomes critical. We're going to talk more about that tomorrow. This is an example of, so in British Columbia, and Washington, Oregon, and British Columbia, we use a matted row system, so the plants grow in the ground, let's say in May. Our first crop is a year after that, and then around for another year. So those plants are in the ground for 3 years. So we have to protect those. We have large aphid populations. So this is up in British Columbia, no aphid control before they harvest their first crop. Lots of plants going down. This is right across the road. Aphid control. So aphid control, and again, this strawberry aphid only feeds on strawberry. So it's a relatively easy aphid to control if you work on it. This is another field up in British Columbia, lots of aphids, no virus. This particular field is on the island where Vancouver Airport is. To the west is the Pacific Ocean. It's the only strawberry field on the island. If he starts clean, there's no virus present. The aphids, you have a few aphids in it, or actually quite a few aphids, but there's no virus to move, so the plants do great. So what changed around 2000? In the Northwest, strawberry crinkle became a big problem for us. We don't know why. In California, the greenhouse whitefly became naturalized and actually displaced the native whitefly, Babesia tabacyi. And as a result of that, lettuce infectious yellows, which was the #1 disease of lettuce back in the 80s and early 90s disappeared because the greenhouse whitefly doesn't transmit it. I mean, you'd be hard pressed to find that now in California, this disease. What precipitated those changes? Most often it's a result of human activity. We probably bought in a lot of ornamentals or whatever it was, but this whitefly came in and became established and outcompeted the native. Climate change, it's hard to go back and say that's the reason. It could be, but more likely it's human activity. It could be climate change. So the other thing we have now is we've got, if we look at strawberry plants in California, so this is whitefly transmitted, that's whitefly transmitted, and we see those in California. We see some of the aphid transmitted. In our area, we pretty much only see the aphid transmitted viruses, these 4. In the Northeast, those are the 2 big ones. So if you're a nursery up in this region, you got to worry about aphids. If you're a nursery in California, you have to worry about whiteflies and aphids. Blueberries. Blueberries the oddball because one virus causes disease. So we've had a few. This is a pretty cool disease if you're not a grower. Virtually 100% of the fruit falls off the plant when it's about 4 millimeters. I mean, this is prior to harvest. That's what it should look like. Early in the season, you see the reddening on the leaves, but they grow out of that. You can see the fruit on the ground here. Here's this plant's the foreground, lots of fruit. This plant, you don't see any fruit. I mean, it's a catcher. It catches the grower's attention. We've seen it in 2 fields up in British Columbia for 20 years. Recently we found it in 1 field in Washington. Almost 100% fruit trap. It moves slowly. It moves very, very slowly. We have a new virus associated with this in 2014. And at this point, it's almost a perfect correlation between virus presence and symptoms. Again, rolling circle, this is Alfredodid, blueberry fruit trap. You cut it, you get these bands. This one is actually a host, but sequence the product, develop primers. No rolling circle in plants that were symptomless. So the virus itself, it's an unusual virus. It's large for other viruses in that family. It's only got 1 large open reading frame, similar to tuning vein clearing, but the way the genes are organized. But the sequence is much more like cauliflower mosaic, likely going to be a new genus. This is blueberry fruit trap, 1 large open reading frame, the tuning vein clearing, cauliflower mosaic. These 2 are more closely related based on sequence. These 2 are more closely related based on how the genes are organized. Correlation between symptoms and fruit trap. So what we did last year is we just picked a row in the field that looked like it had about half plants showing symptoms, and we tested. So I went through and scored every plant for symptoms or healthy. And so based on symptoms, we had 31 showing symptoms, 34 not showing symptoms, and 1 showing questionable. And we had a perfect match with these. This one was positive, and we did the test again in summer. These symptoms are based on springtime, and we did the symptoms again in summer prior to harvest when the fruit fell off. And again, perfect match. Well, this one is questionable because it had another disease that a lot of the flowers died. The other thing we saw is that this is a field in British Columbia that was taken, this block was taken up because of the fruit drought. This field had been there for 20 years. This is about 30 meters. So we tested the first plant on 24, and everyone was negative. These have been in there for 6 years now. We tested the first 24 plants here. Everyone was negative. So again, very slow movement, and it may only, at this point, maybe it only goes to blue drought. We don't know that. So last year I talked to the growers about eradicating it. So they went in, and this is the field in Washington where we did the testing, and we marked every plant. They went in and cut everyone off. I was up there in May, and I said, these plants are all going to still be a source of virus. Cutting them off isn't enough. You've got to actually spray them with herbicide. That was on a Monday. I went back on Thursday before I went back to Oregon, and they had all been sprayed. So they're taking it pretty seriously. The product grain blotch, this is one that only occurs in the southeastern part of the U.S. And this is an interesting one in that every single lesion is a single infection point. This virus does not go systemic. It's also very unusual. It's the only virus we know of with 2 helicases, not plant virus, plant, animal, human, bacterial. It's the only virus we know that has 2 helicases, which suggests that it's probably a very recent evolutionary. We have a helicase here and here. This one is more like a Romaviride, that's like a Tobano. So the helicases came from 2 different virus families, suggesting a recombination event. Flu virus from a molecular point of view. That disease was first observed near Athens, Georgia in 2006. In 2008, they found it throughout the entire state. In 2012, it was over the entire blueberry production region in the southeast. These are other major blueberry producing states. We haven't seen it anywhere else. But the thing is, this virus does not go systemic. If you take cuttings, take the leaves out and grow it out, next year it's healthy. So that means every single lesion you see on that plant is a single separate infection. Viruses and berries, we got a lot of different genera. These with an asterisk are probably where we have new genera found in berry crops. This is just rubus. Tomorrow we're talking about nurseries. We're at the very top stage. We want to test for everything. But again, there's quite a mix there. Strawberries, these are the common ones that we see. Worldwide, these are the most important. These are relatively minor. These are rare, very rare. This is a shock to me because cucumber mosaic is a very common virus in many, many parts of the world. And only recently, in the last couple of years, they found it in strawberry in China, and it's causing a lot of problems. Why now? Why only in China? We don't know if it's an unusual strain at this point. And this is one that I think potentially could be of interest here, in that, not that this particular virus, but it's a Gemini virus. You guys have a lot of whiteflies, you have a lot of Gemini viruses. If you're going to find new viruses in strawberry or berry crops here, I would expect it's going to be a bacteria. Because you've got a lot of potential viruses, and you've got a lot of bacteria. Blueberries, these are the common ones. These are uncommon. This is one we just did recently from deep sequencing, only in New Zealand, only in China, only in Japan, Michigan and Japan. So these are relatively rare, these are more common. But again, when it's a certification program, we start out testing for everything. Virus on the move, we have quarantine to try to prevent these things moving from country to country. We'll cover that more tomorrow because we're running out of time. Certification we're talking about tomorrow, talking about that tomorrow, talking about that tomorrow. Production fields, disease control. If you're a grower, you're worried about disease control. If you have a virus that's not causing any symptoms, you don't really care. So in a nursery, you want virus control. So there's 2 very different focuses on virus control, depending what you're doing. Manage the risk. You know, if there's a risk, manage it. You know what the viruses are. That's not as bad as it looks sometimes. Who, what, when, where, how they live. So that's what you need to be able to answer. What viruses? Where are they living? How do we manage them? Where are they coming from? A lot of different workers, technicians in my lab, graduate students that I work on, collaborators around the world. A lot of different funding sources. These are larger grants. These are all grower groups around the U.S. and Canada. Recent reviews on berry viruses, I can send these to you. Recent advances in strawberry virus detection in 2006, blueberries in 12, and rubus in 2013. I can send those to you, and if people can get them, I mean, it's PDFs. But they're, except for the strawberry one, these are all pretty recent. So, let's keep those numbers. Berries.
Thank you.